Hello everyone. We will be starting Mendelian Genetics as our first lesson, which is section 5.3 for AP syllabus, and it will be covering the objective, explaining inheritance of genes and traits as described by Mendel laws. Alongside AP objective, it will also uh, lay the foundation for achieving objective for MSAT, which talks about complex nature of inheritance pattern in animals and especially in humans. If we look at the contents that we would be covering during this session, it would be a little introduction about Mendel. It would be the reason to understand his choice of organism and how it was so helpful to achieve the laws of genetics, and how the laws of genetics were put into certain mathematical calculations of probability and certain other techniques which we use in genetics to test the genetic combination of a character inside the living organism. Alongside these concepts, you will be finding certain hyperlinks in the presentation, and those hyperlinks will lead the students to further additional visual resources just in case if they require furthermore explanation, and it is also assisted by practice questions so that if there is any confusion after doing the practice, you would know your weak areas. Let's begin. The first thing that we would discuss right now would be Gregor Mendel, an Australian monk who was having a duties of religious nature as a profession, but alongside he had a scientific mind and it was bent more toward understanding genetics. During the times when Mendel was living, it was a common belief that when two individuals, be plants or animals, they get into the process of reproduction, their offsprings show blended characters. There was nothing like a child or an offspring could be like any one of the parent. So Mendel had curiosity about how this formation of characters works, how this genes or anything, because at that time there was no idea of genes, they move between generations and develop certain characters. But since he was most of his time in the church, so he wasn't sure what organism should he use, because he was doing a scientific study, and if he has to reach a conclusion, he needs to convince other scientists of the world by his experiments and by his data. And to do experiments, to collect data, we require a lot of research work and space as well. And he was having one constraint, that he wasn't having much space as a lab for his work. He was having backyard of his church that he could only use. So initially he thought that he's gonna use mice as his specimen organism, and he did try them, but he found out that taking care of a living organism require another level of effort and resources, and the nature of character is more complex. So he came up with a choice of a living organism which is called pea plants. And let me show you a little bit. I did not add the picture of the pea plants there. This, a garden pea plant. He came up with this particular this particular organism. And there were so many reasons for the choice of this organism. The first reason being that this particular plant was having a lot of contrasting pair of characters, which means that there were a minimum of 7 characters that this plant was showing, where there were only 2 extremes or 2 options. There was no blending, there was no third option. So it was an easily observable characters that the plant was showing. To add on for the examples, I would say that let's take height of the plant. So either the plant was showing a 7 feet tall height, or the plant was showing a 4 feet tall height. Either the plant had purple flowers, or the plant was having white flowers. There was no third combination. Another important reason was that they were so thin and they require so less space that even in the backyard of the church, he could grow somewhat plants between 700 to 900, which is a good amount of data record for one generation. And being a plant and very simple in its life cycle, Mendel could get 2 generations in 1 year, which means that if he worked for 6 years, let's suppose, so he got a record of 12 generations minimum, and then every generation involving 900, 700, 800 individuals, which was a considerable number, and any result based on this amount of data would be counted as credible data, and no one would raise their judgments about it. So for this reason, the choice of pea plant was a very wise choice, and taking care of them was easy. They were growing fast, and these pea plant study gave us the foundation of three very important laws: law of dominance, law of segregation, and law of independent assortment. And these were the three laws that made the foundation for the modern day genetics. Before we move on to understanding what these laws are, we need to introduce ourselves to certain key genetic terms, and the first term being pure breeding. You need to keep one idea in your mind all the time, that for any particular character in the body of a living organism, there is a pair of genes involved. And why we call them a pair? Because we receive one member for that particular character from one of our parent, and the other member from the other parent, because this is the basis of sexual reproduction, that the two individuals contribute the same characters, but each of them contribute half of the information. So when it combines in the individual, it gets the full information for that particular person or organism. So if I say that this particular tall plant is pure breeding, it means that for the character of height, the genes that he got, that this plant got from either of its parents, were completely similar, right? And the short plant got the two genes from the parent. They are exactly similar to each other, which means if I show the genes with alphabet, so I would say that this plant was having, and this is the strong character, which means that most of the members, one criteria is show this dominant character. So let me say that this was having capital T, and this was having gene combination small t, small t. Other than being pure breeding, the other important definition that we need to keep in mind is the word traits. Wherever the trait word is used in genetics, it means we are talking about characteristics, which can be physical characteristics, it can be any internal characteristics as well. So Mendel basically studied seven main important characters in the body of the pea plant. The other important definition is allele, which is that each member of the gene in a gene pair is called an allele. They can be similar, they can be different. For example, a gene pair can have both the genes capital P, or small p, or they can be in a combination of capital P, small p. So in any of the situation, we would say that these two genes are alleles, which means we are talking about one character, but we are talking that the purple color and white color are the two options within one character. So these two are alleles to each other. In the same way, if I go back to the video, so we would say that the gene for the tall height has an allele which is controlling the character of short height. The gene for the green color of the seed has an allele which control the production of yellow color of the seed. If both the member in a gene pair are similar to each other, we would say that this is a homozygous condition. But if the pairs are different from each other, we would call it a heterozygous condition. It is the heterozygous situation which actually creates a lot of different surprising results. Another important technique that we need to understand before moving on is the Punnett square construction technique, which is a technique provided to us by a scientist called Punnett, which is a diagrammatic way to predict the combination of genes that would come up in a generation. So, which means that if I know the genetic combination of the two parents, so I do not need to actually put them into an experiment and wait for the offsprings to come out. I can do it on paper by using those genetic combinations of the parents to predict the genetic combination of the offsprings. For example, I have a parent here with yellow color and is showing a dominant characteristic, and a green color seed by a plant which shows a recessive character. Both of them are homozygous. From this point onwards, we will be using key genetic terms to explain our question. So we can, and then we need to understand that each member in a gene pair during the process of formation of male gametes and female gametes gets separated from each other and they go into individual gametes. So we will write the gamete for one of the parent on one side and for the other parent on the other side. It's totally up to you which parent you choose to put on which side of the grid. Secondly, you can, just to show your additional strengthened concept ideas, is that you can show how one of the parent is male and the other of the parent is female. You can use their symbols as well. So if we have to see it, we need to understand that this particular A has the chance of combining with a small a, and this particular gamete has the chances of combining with another recessive allele over here. So as a result, we would get 4 individuals. All will be heterozygous because they are receiving one dominant gene from one parent, second recessive allele from another parent. So apparently, maybe they are showing the character of the dominance, but internally they are not pure breeding, they are heterozygous. So you will be putting a lot of practice of the Punnett square once we are done with the examples. Law of dominance is based on a practical or a research that Mendel did for 7 different characters, and every time when he grew the plants, he studied only one character at a time. He did an experiment by taking 2 pure breeding varieties for a particular character, let's suppose height, and he crossed them together, taking one as a male parent, taking another as a female parent. And he figured out that when these plants, they produced seeds, and those seeds were grown into first generation, these plants, all of the 700 to 900 plants, showed the character of one of their parent, and the other parent characteristic was completely missing, which was surprising for Mendel. So what he did, that where did the character of the other parent went, he took the 2 individuals of the same F1 generation and he crossed them together, and he figured out that in the second generation the characteristic of the grandparent appeared, but in a very limited quantity, but it was there. This led Mendel to believe that in every character there is one form of the gene which is so powerful, which is so strong that it can mask the effect of the other gene. or it can dominate the appearance of the other character. But when we repeat them and we bring them down to second generation, somehow the recessive allele from each parent gets combined and show their impact in the second generation. This is called law of dominance, that when a pure breeding tall plant combines with a pure breeding dwarf plant, the resulting heterozygous individual apparently show the tall parent character, but genetically they are carrying the gene for both the parents. And when both the same heterozygous are crossed, they show the character of the second parent but in a very limited quantity, which means that whenever there will be a dominant gene, it will not allow the recessive gene to show its impact. He not only, just to convince people, did this experiment only to study height of the plant, he used all these characters, and in all these characters he figured out that one of the form was dominant, one of the form was recessive. One of the form was dominant, the other form was recessive. So whenever there was a cross, the F1 generation only showed the character of one of the parent. Never in these 7 characters, which he planted the plants separately, he could not find that in F1 generation both the parental characteristics appeared, which means that the characters are clearly sorted out in 2 categories. Either they are dominant or they are recessive. But in the second generation, the recessive character appear, but in a very limited quantity. And if we put it to the ratio, we would say that it was in a ratio of 3:1. And this 3:1 is also sometimes put up as law of segregation. Law of dominance and law of segregation has very little difference, because they practically speaks about one character and the inheritance of that one character over 2 generations. So if we just see it from another perspective, we would say that there is this tall plant which is pure breeding, there is this dwarf plant which is pure breeding, and when we are going to develop gametes, so I'm only showing one gamete, the other gamete is there which will carry the second pair of the gene, but we are missing it over here just for the ease of understanding. So what is happening, that only one member of the gene pair will travel in one gamete. When they are going to combine, so this particular individual, which is a child to these 2 plants, will receive a member of the gene pair from each parent. So this is a heterozygous plant, apparently showing the character of one of the parent, because this capital T is so strong that it would not let the small or recessive character to appear and come on surface. Now, on the other hand, if we plot this 2 heterozygous plants over here, this heterozygous plant as one parent, and we choose another plant from the same F1 generation as the second parent, so we would find that the 4 individuals that are produced are in a combination, which is 3 of them are tall and 1 of them is dwarf. The 3 of them that are tall and 1 is dwarf is counted as the phenotype ratio, which means that apparently when we look at them physically, 3 of them shows the character of the dominant parent and 1 of them shows the character of the recessive one. But when we look at the genetic combination, we find out that 1 is homozygous dominant, 1 is homozygous recessive, and 2 are heterozygous dominant. So the genotype ratio would be 1:2:1. How is it different than the law of dominance? You would say that law of segregation talks about transmission of one character at a time from one generation to another generation. At this time, We did not talk about any other character. What is the color of the flower? What is the position of the flower? No, we only focused on one character at a time. Law of independent assortment is a little complicated because over here we took two characters, we studied the two characters and we took them together from one generation to another generation. So the law of independent assortments say that when you take two characters at the same time from the pure breeding variety, so does it happen that the gene pair for one particular character, does it affect the transmission of second pair of gene or not? So that was the basic understanding. And when the cross was carried out, the gametes were created. One member of each gene pair would go into the gamete. It resulted in a hybrid individual, again showing the dominant recessive law, and it showed both the dominant characters from the parent. And when we took two heterozygous and crossed them, we got a bigger Punnett square and we got 16 individuals going for the formation that 9 out of 16 were showing the dominant characters in both the situations for seed color and seed shape. 3 out of 16 showed 3 out of 16 showed a recessive character and a dominant combination, which means round seed with green color. 3 showed wrinkled seed with yellow color, and 1 out of 16 showed the recessive characters for both the characters that we were studying. You will have a little difficulty over here for creating gametes. You can use FOIL method for creating gamete. FOIL method means, which is very easy, you take the first gene and you take the first gene for the second character, and then you make that capital Y capital R, then use the first gene in the first gene pair and mix it with the second of the second gene pair, so capital Y small r. Then you have the second gene in the first gene pair, combine it turn by turn with another gene pair. So you would get the gametes, which is not that tricky, but sometimes students, they find it confusing. And the idea and the results from this law basically stated that when two gene pair are traveling from one generation to another generation, they do not have any impact on each other. They are going independent of each other, which means that this particular strong gene would not ask the strong gene of the shape of the seed to be with it. You will not find that capital Y is always pairing with capital R, or small y is always pairing with small r. There are all sorts of genetic combination. They are going independently. There is no influence of any character over the other. I think this is enough of the discussion about the 3 laws of genetics. I have given a couple of practice problems. You can see these questions and you can pause the video at this time, because in the upcoming slide you will have the answers to these questions. So there are steps that I have given to those who really want a little help how to create a genetic diagram. So follow these steps. For any question that comes in front of you, write down first see what character are they talking about, then choose a symbol, which alphabet are you gonna use, then decide are the parents in the question homozygous or heterozygous. And once you have made these 3 decisions, write the 2 parental genotype combination, make a cross between them, and set up your Punnett square. So this is one of the Punnett square combination that I have provided. A plant with yellow seed is crossed with a green seed plant. What percentage of the offspring will be yellow? So write out the symbols for the alleles, write down the genotype, write down the cross between them, and construct the Punnett square to predict the offsprings. These are the two problems. At this point, I would like you to pause it, read these questions, and construct a Punnett square, parental diagrams, or anything, and practice it. And once you are done with your question, you can resume the video to see the answers that will come up now. Okay, so these are the answers to the two questions. I hope that there won't be um, any confusion in constructing these genetic crosses. Just in case it is there, provide me the feedback. Where was the basic problem? Which step was really hard to construct? I will be creating a second part of the video. This is the practice for the dihybrid cross. Pause the video at this time as well. Create a dihybrid cross, and you have the answer for it provided in here. So this is the checkerboard that would come for your dihybrid cross. Practice it and see what combinations you come up with. I have the answer over here, and then we have the law of probability, which I will be recording separately alongside the test cross. Till that time, please give me the feedback. How was the answer? Which part of the genetic problem was creating trouble, and which require further more assistance? Have a good time. Have happy learning. Bye for now.